So welcome everyone to our Cheminformatics meeting. I will just give a 2-minute or so introduction. Afterwards, I will hand over to the speakers of our program for today. So what are we trying to do here? So the aim of those meetings is to bring cheminformaticians in the widest sense, so modeling, bioinformatics, machine learning, together scientifically and personal, that people meet as well. And so we originally had personal meetings, physical meetings in Cambridge, and that was 30, 40 people. And right now we usually have a few hundred people registered from all over the world. So it really makes those meetings much more accessible. And I think it's great that people from, I don't know, Japan to Brazil or so are joining here. So welcome everyone. And so what are the future dates? And so mark them on the calendar. And please let me know if you would like to present as well. In particular, we're always looking for speakers. So the last meeting this year will be on the 24th of November. And for next year, I will set the dates shortly and circulate those. And there's this c-inf.net, that's our event website. And there's also a Cambridge Cheminformatics newsletter where I'm circulating events and jobs and so on. And so you can go to the drugdiscovery.net website. Also, if you're inclined to do so, follow me on Twitter if you wish. So the main meeting, as I just said, it will be recorded as agreed with the speakers, or those speakers who agreed to record it. And afterwards, those presentations will be put on our Cheminformatics YouTube channel. So in case you're interested in particular topics or the time zone is unsuitable, you can give those recordings a look afterwards. As for questions, so please type your questions in the chat window. Feel free to do so also during the presentation. And afterwards, we'll go through them orally as time allows. But feel free, we can collect questions already during the presentation as well. And afterwards, the speakers, if there are questions remaining, would be great if you could be so kind to follow up by chat for the remaining questions as well. And yeah, important. So it's partly about science, right? But afterwards, it's also about meeting people. It's easier in person, of course. So put your beer or whatever drink you prefer into the fridge, because afterwards what we will do is we will put people into a random breakout room, so 5, 6, 7 people together, so you can have a chat with either people you know already or make new friends from the cheminformatics area as well. So please stay on afterwards, after the third talk as well. And yeah, if you wish to present at the next meetings, please let me know, as well as if you have information to circulate, so I can include them in this Cheminformatics newsletter. It's 300 people or so maybe. And so it's a good way to reach this very specific group of people from this area of science. Yeah, and so that's the program for today. So 3 presentations. First, we will talk about computational prediction of allosteric sites in GPCRs by Irina Tikhonova from Queen's University, Belfast. Then we will have DECIMER, so deep learning for chemical image recognition by Steinbeck from Jena. And finally, probabilistic random forests for target prediction by Marianna Trapotsi as well as Mervin. Okay, and so with this, I would like to hand over to our first speaker, Irina. Please, I will stop sharing here. And so you can share the slides and feel free to go ahead afterwards. I think you should be able to start sharing.
So yes, Andreas, thank you very much. And so thank you for invitation to give a talk in such a wonderful meeting, a very famous meeting. And I'm going to present one of our recent projects. And this is the project where we have developed a computational protocol to predict allosteric sites in G protein coupled receptors, or in short, in GPCRs. And first, let me give a little bit of background around those receptors. Just one moment. Okay. Oh, sorry. So G protein coupled receptors, I guess everyone knows, is the largest membrane protein family, and there are more than 800 members in the human body. So GPCR transduce a signal inside of the cell from a variety of endogenous ligands, and as a result, they have broad influence on human physiology. So 475 GPCR drugs are approved by FDA and around 300 in the clinical trials. So although GPCR drugs have shown substantial therapeutic success, developing drugs for many GPCR subfamilies have proved to be challenging, and the challenge is due to difficulties to achieve selectivity when you're targeting highly conserved orthosteric sites where endogenous molecule binds. So we also know now that there are many small molecule ligands that can modulate GPCR by binding to the sites which are distinct from orthosteric sites. And in simple classification, those allosteric modulators can be divided by as a positive allosteric modulators that sort of enhance binding and signaling of orthosteric agonists, and also negative allosteric modulators, so-called NAM, that actually in opposite reduce activation by endogenous orthosteric ligands, or just endogenous or just simple orthosteric ligands. So allosteric modulators have a number of advantages compared to orthosteric drugs, and this slide summarizes it to you. So allosteric drugs have a better potential for receptor subtype selectivity due to greater sequence variability in allosteric sites. They confer probe dependence and functional selectivity, causing selective receptor activation and thus different tolerance in chronic diseases. They have also diverse relationship between duration and intensity of the effect, so which can prolong the therapeutic effect and without drug overdose. So currently, 2 allosteric modulators have been approved as drugs by FDA. So despite the clear potential benefits of GPCR allosteric modulation, so the discovery of allosteric drugs or allosteric modulators sites as well, allosteric sites and lead compounds have been mostly, I guess, random. So through random screening of compound libraries. And this was till recent moments, and so now the situation is changing, and with all those brilliant achievements in structural biology of GPCRs, so we now have a number of structures of GPCRs bound to allosteric modulators. So these are crystal structures or X-ray structures or electron microscopy structures. And here I show you a summary of those structures. And there's a space filling representation in green. You see the location of allosteric modulators in the GPCRs. And these structures indeed show this remarkable diversity of locations. So this allosteric site can be seen at the extracell site or intracell site within the helical bundle of a GPCR, or actually we also see allosteric sites at the interface with lipids, and those at the interface sites also have different locations. So these structures provide the first insights into GPCR allosteric modulation by small molecules, and of course create this fantastic ground for development and validation, new computational tools for prediction of allosteric sites for other members of GPCR family, or in fact for all membrane proteins. And this is a good role model to follow here. So, basically another thing that we can learn from these structures, that lipid bilayer plays a substantial, seems like, role in the formation of those sites, and especially if we see those sites at the interface. So what it means, accounting for the receptor in realistic environment and dynamics of the receptor dynamics in this environment is particularly important for accurate prediction of allosteric sites. So, and the way how we can do it, so the most, I guess, widely used tool to look at this crystal structures and give a life and look at the realistic environments, of course, to apply molecular dynamic simulations, which have been proved to be successful. And among those methods, cosolvent molecular dynamic simulations could be very interesting to consider, and these methods have been largely used for soluble proteins. And within these methods, we use these small probes, as you can see here on the right side, usually organic solvents like a pyridine, acetamide, isopropanol, to map the sites. And those probes can quickly diffuse into those cavities, free cavities in the protein, and estimate the accessibility of these cavities, and therefore predict its potential binding cavities for larger molecules and very efficient manner. And so covalent mapping simulations have been really successfully used, as I said already, for soluble proteins. And there are only few studies we see in the literature application of these methods for membrane proteins. And this is, of course, if you think about this and you start to do it, you will face a lot of challenges to apply covalent MD simulations for membrane proteins. And here at the bottom I summarize, so it could be a limited probe sampling, since we are dealing with large systems, probe non-specific binding, accumulation probes in membranes. Also, this issue of membrane distortion. We know from literature that membrane can tolerate only concentration of co-solvent, organic solvent around 5%, while we know also from co-solvent simulation of soluble proteins that we need to keep around 10–20% of the solvent, co-solvents in the simulation to achieve successful mapping of the binding pocket. So, and what we are trying to do is we're trying to basically bring this covalent MD simulations to the world of membrane proteins, and we try to develop protocol that allow to do this. And our protocol has the two key features that kind of try to address those limitations. So first is that to avoid this membrane distortion and penetration to membrane, and to increase the sampling around the protein at the same time also with a reasonable concentration. So we introduced so-called cylinder-shaped harmonic wall potential, and here I show one of the example. And so this potential, this restraint allow us to sample specific area within the GPCR. For example, here you see extracellular site, and we can do the same for intracellular site, and also define area at the interface with lipids. And so applying this restraint, what it means that so when the probes reach the wall of the cylinder, the repulsive bias applied so that it pulls back the probes, and probes actually really stay around the protein and have high probability to basically interact with the protein and not diffuse to the distant water layer or to the lipids. And the second part is to enhance the second feature that we introduced to enhance specific sampling. We propose to use the fragments that in the simulations that derived directly from the structure of GPCR allosteric ligands, and just one of the example. And now I just want to give you more details about those features and how these features were implemented and tested, validated on example of G protein coupled receptors. So this slide shows you basically the protocol that involves system setup, system equilibration, and system production. And in the case of system setup, so we define this protein, we define the box, a probe water box in the user specified place, either an extracellular site. I don't know if you see my mouse or not, on the intracellular side. We also allow to include or exclude in opposite orthosteric ligands. In the case of mapping site for positive allosteric modulators, as I will show later, it's important to keep orthosteric ligand because it stabilizes the allosteric site. What will be example? So once we set up the system and we embed it in the water lipid bilayer, so we have all atom simulations here. So we run equilibration, and within this equilibration, the probes actually confined in this cylinder that you see on the top, we don't allow to interact with the protein. And once the system, overall system is equilibrated, we actually then go for production. And within production, we remove the boundary that faces the protein, and the probes can actually move towards the protein, interact with protein. At the same time, we also extend those walls of the cylinder. As you can see, now we have longer cylinder, and the probes can basically confine within this cylinder and thoroughly map, in this case, extracellular site. So Production step runs 40 nanoseconds, and with such setup we can use 10% concentration of the probes. What also important to say here, so we propose to use separate protocols for sampling, for example, specific area of the GPCR, so to reduce number of restraints. And for the case of mapping interface sites, so at the receptor lipid interface during production step, we introduce two collective variables that define specific area of the GPCR. So, and this is example you see here for purinergic P2Y1 receptor, where the site locates around the helix 1 and 3. So, and we basically ask probe to actually sit within this part of the cylinder and sample this area. So, okay, and now basically you see hopefully the main idea of this protocol, and we test this protocol using 3 case scenarios. So we choose muscarinic receptor with allosteric site on the extracell side, and this is a site of positive allosteric modulator. We choose beta-2 adrenergic receptor where the site locates on the intracell side. It's a negative allosteric modulator, and we choose also the case scenario for interface sites. It's a P2Y1 receptor. Also, the beauty of these case studies is that we also have crystal structures of those receptors where we don't have allosteric modulator. So we can actually assess how this allosteric cavity, not kind of pre-shaped by allosteric modulator, can behave in those probe MD-based simulations. And for these case studies, we, as I mentioned before, introduced a second feature. So we identify specific probes. And we know from the literature that, you know, when researchers analyzed the database of allosteric modulators, not only a GPCR, but for different targets, and compared with the orthosteric ligands, they found that those allosteric ligands have a couple of distinct features. So they have more aromatic atoms per heavy atom, they have few rotatable bonds, so they're rigid. So we kind of thought that really it maybe would be more suitable to take probes, probe molecules that derive directly from the structures of allosteric modulators, so that we can actually hopefully to reach better sampling and specificity. And now I show you in the case of muscarinic receptor, there are a number of ligands allosteric modulators. So we took a common substructure of allosteric modulators to derive the probes, specific probes, we call them. And in the case of beta-2 adrenergic receptor and P2Y receptor, we have only one ligand, and here we use fragmentation to get specific probes. And this table at the bottom now, you see our specific probes that we used to run this probe confined simulations, and specific probe confined simulations for these 3 receptors. So eventually we run these receptors with at the answer 5 probes, so 2 specific, 3 standard probes that you've seen at the beginning of my simulations, and we use 2 conformations of receptor. And now here I show you the actually trajectories, the way how they look like, so that you could appreciate, and for each of these receptor, and this is, I choose specific probes to visualize the results, and also for P2Y1 receptor, and you see, so the probes, they don't aggregate, they move, they actually reach the site, they hold in the site, and they form interactions. And of course, the most fascinating site at the interface, and you also see that the probes actually can reach the site. So I visualize residues that form the site in this stick-like representation, and you see space-filling representations coming from the probes. So we generated quite a large number of those trajectories, and we, of course, then wanted to test it and quantify this trajectories. And we did two types of analysis. So we did retrospective analysis of those trajectories, just to basically appreciate the probe presence in allosteric site. We calculate probe occupancy. We also tested in a settings of predictive analysis, assuming we don't know where the allosteric site locates, and then we engaged with probe density, cavity detection, and residue probe interaction energy analysis to actually see if the probes can identify the key residues of the allosteric sites. And now I just give a little bit of information around each of these analyses for 3 cases. So retrospective analysis, so we calculate the probe occupancy, and so it's basically how probes sit and interact with key allosteric hotspots, and those hotspots defined, as you see at the bottom, based on the residues that form strong interactions with the ligands in the crystal structure. And you see that we run lots of systems, and in the case of muscarinic receptor with agonist bound, and then also empty form of the receptor, we also, you see all these 5 probes, and also this data based on the multiple trajectories, aggregation of multiple trajectories, yeah. And also you see here beta-2 adrenergic receptor, the occupancy table, and for P2Y1 receptor. So the kind of key points, what we have learned from this retrospective analysis, that indeed specific probes work really better compared to standard probes. So you see in those highlighted in bold, the occupancy in the case of specific probes is high. We also see in the case of muscarinic receptor, it's really important to have this orthosteric agonist in the binding site, because we see better occupancy. So and it has been shown previously in classical MD simulations that orthosteric ligands stabilize allosteric sites. So it's good to consider in the simulations, co-solvent or probe simulations, orthosteric ligands. Also, what is interesting to see that both conformations can actually catch those key hotspot residues. So either the one that produced, I guess, taken directly from the X-ray structure bound allosteric modulator, or the most interesting, of course, from crystal structure where we didn't have allosteric modulator. Even though some of the results for this second conformation a little bit lower, and so we don't map one of the sites. In the case of beta-2 adrenergic receptor, you can see those zeros. So, but still we can map a number of key residues within the allosteric site. So next analysis that we have done, as I said, predictive analysis, and in this case I just want to focus on purinergic receptor at the interface, the most interesting, also a challenging situation. And what we've done, so we basically aggregated all those trajectories of probe simulations, and we calculated probe density, and here is just visualized based on ISO value 5. So we use a wall map of a VMD program to get those probe densities. And from probe densities, if we would just extract residues that actually close to this probe density, so there are 27 residues that potentially could be part of the allosteric site. But we also recognize not all kind of areas where the probe resides is actually cavity. So therefore we combine this analysis with cavity detection kind of analysis. So, and cavity detection here was done with MD pocket. So this program allows to monitor cavities with an MD simulation trajectories. And on top of that, we also, together with F-pocket, MD pocket can actually predict druggable cavities. And in this case, you see for P2Y1, we actually have only druggable cavities within helical bundle, and cavities of our interest, which is at the interface, is not druggable. So, and we see this as scenarios for other also GPCRs where we have a site at the interface. So in this case, especially in the blind search, if we're searching for cavity, of course, we need to look at all detectable cavities. And in this case, what we've done, we basically overlap those results from cavity detection and probe density, and we identified the area where there is this overlap. And from the overlap, we also managed to reduce number of residues. So it was 18 residues in this case. And so those residues were used for probe residue interaction energy calculations. And when you do it, you immediately can get the list of residues and really a few residues that actually form strong interactions with the probes. And those residues will be kind of putative residues that form the putative allosteric site. And here you see that highlighted the residues that actually form interactions, in this kind of bold, form interactions with ligands, and they ranked pretty high. I have to say, in the case of muscarinic receptor and beta-2 adrenergic receptor, the results even better because there we have number of polar interactions, and the cavity for case of M2 was predicted as a druggable, for beta-2 half druggable and half non-druggable. So, but what we can learn from this, and that's key points, that the probe binding interactions are matched with allosteric modulator interactions in the X-ray structure, and not all allosteric cavities at the moment as predicted as a druggable using criteria that we, for example, in this case we use F-pocket, and all and in the blind search, if we have this situation, we need to look at all detectable cavities, and in this case, of course, the probe simulations can help really a lot because they could assess the accessibility of this cavity and also polarity of this cavity. And this is particularly important for the sites at the interface. Okay, and so what also here I would like to mention, we made another test for P2Y1 receptor where we use those specific probes and sample other interfaces, is not only the one that I show you at the very beginning, and to see whether those probes actually go to other interfaces or they actually stay specific to this particular cavity. And we calculated occupancy, and as you can see that either there is no occupancy, so the probe doesn't simply go within the specific interfaces, or the occupancy is pretty lower. So from this little exercise, we also could say that the probes that derived from structures of allosteric modulators could be quite specific in identifying distinct cavity, even though, of course, we probably need to test more here and take more examples. And also what I want to show, the most interesting part of this project is that we actually went even further and we did truly predictive validation of this protocol by predicting a binding site, allosteric site for this positive allosteric modulator UCB compound that was identified by high throughput screening in a dopamine 2 receptor. And so we have crystal structure of dopamine 2 receptor. And indeed, this is quite a nice starting point to run those simulations, cosolent simulations, and identify allosteric site. So we fragmented these ligands and we run those probe simulations, and here you see probe density analysis, and we looked at the different scenarios, extracellular, intracellular, and also interfaces. So all cavities that were detected by MD pocket was sampled by probes, and we already see that quite a lot probes reside on the extracellular side. And the MD pocket results also show here that there are lots of different cavities, and one of the cavity actually at the extracellular site is predicted as a druggable. So it turns out this example was pretty straightforward in the selection of actually putative cavity, which we selected. And then we also have done docking of this molecule, and the poses were selected based on the based on the probe simulations. And from all this kind of multiple analysis, we selected 6 residues for site-directed mutagenesis, and this pharmacology was done by our collaborator and his group, Peter McCormick from Queen Mary, University of London. And out of 6 mutations, we identified that 4 mutations really affected this positive allosteric modulation of this compound on dopamine stimulation of the receptor. And the other 2 were actually potentially also important, but they affected the binding of dopamine itself. So it was not really a very clear-cut results then to interpret. So currently we test this protocol for a number of GPCRs, and we look at even more sophisticated cases, as of course at the interface. And of course, if you have the question and if you're interested, if you have interesting allosteric modulator and a GPCR, and you want to map allosteric sites so that you could then basically run this optimization MedChem program around this molecule. So we are happy to help to predict this allosteric site. So to conclude, the key messages here that there is this quite efficient protocol based on ND simulations that consider also the impact of lipid bilayer on allosteric sites. And so with having those features that I have mentioned, this wall potential that we introduce, those restraint simulations, and also specific probes, fragments that derived from structures of allosteric modulators, we can actually overcome those limitations that I mentioned around the cosolent mapping protocols that can for membrane proteins, and so use it successfully for prediction of allosteric sites, not only in GPCRs, but other membrane proteins. So it's a quite efficient method and feasible solution to actually initiate rational search of allosteric sites and then structure-based design of allosteric modulators in GPCRs and potentially other membrane proteins. So, and acknowledgements. So I would like to acknowledge people from my group that contributed to this project, and the main contributor, Antonella Cincetta, Marie Curie fellow who was in my lab, and now she runs her own independent lab at University of Ferrara, and my collaborator from Queen Mary, University of London, Peter McCormick, all funding agencies and computational facilities that help us to simulate all these multiple simulations. And thank you for your attention, and if you have questions, I'm happy to answer. Andreas, we can't hear you.
Sorry, I didn't press the button properly. Okay, thanks a lot, Irina, for your presentation. There are a couple of questions already, and feel free, let's do them in order, and feel free to unmute yourself so there's a bit of dialogue, because maybe you want to explain them more. So Saravanan, I don't know if you have a microphone, otherwise I will read it, but feel free to ask your question yourself, Saravanan.
So yes, is it 40 nanosecond production run is enough or not? So it's enough, it's in the case studies that we simulated enough, especially if you sample extracellular, intracellular sites. But for example, we already see for interface, especially if site is really at the center of the lipid bilayer, so we need to simulate longer.
Mm. Okay, and Andrew, are you there? Because feel free to explain your question as well.
Yeah, I'm here.
Oh, yes.
Hi, I'm Andrew.
Hi.
I'm a PhD student in Andreas's lab. I wanted to ask whether you had explored simulations with different constituents of the lipid membrane, because I know there are some sites, there are some GPCR sites which are known to specifically bind particular lipids, and I thought that might, you know, you might be able to validate more if you explored.
Yes, this is Andrew. This is a great question, and I have to say it's the next step for our protocol. At the moment, we just use POPC standard simulations, but we already are running POPC cholesterol and also combination of different lipids, because indeed we recognize that. I mean, we actually see we've done a couple of. We run several GPCRs with different interface sites, and we see that these, the allosteric site stabilize differently in the presence of different lipids. Just in simulations we see it, and we definitely want to include it. But at the moment, I guess it will be next step.
Well, that's really interesting. Thanks. I'd be interested in talking more about that sometime.
Sure. Yeah. Feel free to just get in touch with Irina, I guess, to follow that up. Yeah. And there's one more question as well by Jerome.
Yes. Hello.
Hello.
I was wondering. By the way, interesting presentation. Really enjoyed it. I was wondering, how did you parameterize the probes you use for the MD simulations?
Okay, so yes, for parameterization, okay, so we use, I mean, it depends. So we use either, I guess, the website, Charmgue, or we've done it ourselves.
And what do you mean you did it yourself? Did you use some built-in parameterization tool? And if so, could you use as an example, or did you use computational chemistry to come by those parameters?
Well, yes, so we used computational chemistry tools. Yes, we didn't write any of, I guess, specific programs for parameterization. So, and so I have to say that this kind of work, part of this work will be soon hopefully accepted for publication, and the protocols will be available, so you could see all details. Hopefully it will happen within a very short time, because now it's under revision, the manuscript. So and we open all scripts for everyone to use.
Okay, thank you very much. Thank you.
Glad to hear that. And let's do one final question by Akash as well. Oh yeah, in case you cannot read that, what can be the best?
Hi,
oh, yeah, okay,
I'm there. Can you hear me? But I'm in the middle of the road. I'm walking in Milan. So I was just asking. I'm just curious to know the percentage of POPC and CHOL if we want to prepare a mixed bilayer model for this kind of GPCRs simulations. Is there any? Have you ever tried and got a very good percentage if we mix it? Like, I'm just curious to know.
Yeah, thanks. So I would say it's recommended, and we try 10% cholesterol and also 20%. So I think in experiments they usually use 10% more often, but I would say recommendation would be 10 or 20% actually, because if it's 20, then you have more cholesterol, so and you have more chance basically to see cholesterol around those sites. So because the program will put randomly those the cholesterol molecules, and if it's a small concentration, it might be that the cholesterol will be far from the site, but it's actually it should be important. So you probably want to kind of a little bit artificially increase the concentration of cholesterol, that just to make sure that it's around the critical area.
Thank you. You have solved many of my problems. Really great. Thank you so much.
Yeah, that's great. And is it, Akash, so you're on the streets of Milan, you said right now, yeah?
Yeah, I'm in Milan.
Yeah, no, very good. Yeah, we didn't have that before in the Cheminformatics meeting, you see? That's what's possible right now. Okay, and Saravanan asked whether preprint is there. Let's take that as the last question. Yeah.
Well, yes. So I would think that it will be soon available. So as I said, like we just have a minor revision, and so we're going to submit this really in a few days, and then let's see when it will be accepted. So I guess, hopefully quickly. And then, of course, we can provide pre-instance all the valuable files of this work.
Okay, great. Thanks a lot, Irina. Excellent presentation. And thanks a lot to everyone asking questions as well. Thank you.
Thank you, Andreas.
Yeah, and let's go to our next speaker then, who is Chris Steinbeck from the University of Jena, who will talk about using deep learning for chemical structure recognition. Chris, feel free to share your screen, please?
Yeah, thanks, Andreas. I appreciate and second your remarks about how great it is that so many people can participate in those meetings that we had in person earlier in Cambridge. But I have to say, I also miss the Prince Regent and the Penton and all the nice pubs around your place. I'm going to talk, if I can find my slide, about work that was done by Kohulan Rajan, who is a PhD student in my group, on deep learning for chemical image recognition. Kohulan is, I think, participating in the meeting today. And this was also done by, or co-supervised by, our good friend and collaborator, Achim Zilesny, who you see on the upper right corner of this slide here. Now, let me go back a few years to the motivation for this work. I was deeply inspired by the achievement of Google's DeepMind team to develop the deep learning engine AlphaGo, who beat the best human player in the game of Go, Lee Sedol, in those days in 2016 in 5 games, and actually won 4-1. This was an achievement that was not thought to be possible in recent days and was probably still decades away, but in fact it happened in 2016. And one of the amazing achievements, not against Lee Sedol, but just a few months later, was that DeepMind was able to devise an AI system supported by a Monte Carlo search engine, which would start just with the basic rule sets of Go, just 4 or 5 different simple rules, nothing else. And by just playing against itself, it would discover basically thousands of years of Go knowledge, and eventually surpass human playing strength after just a few tens of hours of self-training. And that's just an absolutely amazing achievement. And as you know, probably a couple of years later, Google DeepMind made a big impact by publishing AlphaFold, which was attacking the problem of predicting protein folding from the protein sequence, which is a totally different problem because unlike here, there was no unlimited training data available. So when we were discussing this achievement over dinner, we realized that obviously the key to AlphaGo Zero's success was basically the availability of unlimited training data through continuous self-play. And that is, of course, quite a rare situation. In cheminformatics, we usually have quite limited training data, and that's a situation also that we need to change. But there are some areas in cheminformatics where we are in a similar situation. In this self-play in Go, basically the rules of the system were perfectly defined, and they were easy to define. So this self-play could happen in an encapsulated universe of rules, if you wish. And of course, also during the development of the deep learning for AlphaGo, algorithmic and hardware improvements also made it possible actually only to achieve this this great success. So we thought that in cheminformatics, wherever we can generate unlimited amount of training data, artificially, and this simulation can be good enough to solve real-world problems, then we can use similar training principles. One area that we are interested in is getting data out of the literature again by basically automatically reading chemical articles. And as you all know, those articles consist of both graphics and text. And if we could produce realistic chemical diagrams through toolkits such as RDKit, CDK, and others, we could then also train a neural network with almost unlimited training data. Now, the structures that we need for this are available in great numbers. PubChem alone, I saw Evan Bolton on the list of participants here. Hello, Evan. PubChem alone is 100 million structures and therefore 100 million images. My group works on structure generators, which can potentially go to 10 to the 60 and more, depending on the molecule size and the atom set that you are using. So here we can generate a lot of structures, and we have been working on a depiction tools done by my PhD student Otto Brinkhaus, who's also on the in the audience, and this toolkit can generate an almost unlimited amount of data. Why are we interested in this? I already mentioned it. We are doing natural product cheminformatics and chemical information. We are dealing with articles like this here, where we have chemical structure diagrams on the right side, and a lot of information about those structures in the text, such as the species from which it has been identified, biological activities, chemical classes, chemical names, and so on. And we want to get this out again, and we want to go back to the 1970s, the 1960s, and rediscover all the information that's in the articles. The problem is we are dealing with scanned pages. We do not have properly annotated text and vector graphics. This is just black and white pixels on the page. Now, in order to do this, before we actually do some nice deep learning and image recognition, we needed to cut the journal pages into small pieces and extract the individual pieces of information. And here, this slide is about the DECIMER segmentation tool, which was done by Kohulan in collaboration with Otto Brinkhaus, who is here depicted on here at the bottom. And this tool is also available as a web service, and with a web interface. You can look this up in the article quoted below. And the tool has become really good in getting structures out of the paper so that it can be fed into our image recognition system. Now, once you have these structures, then you are facing the problem of retranslating this collection of black and white pixels into a machine-readable representation. There are algorithmic solutions to this, such as the OSRA package developed in Mark Niklaus's group. But we were interested in really using the deep learning methods that have recently been so successful in cases like AlphaGo to solve the problem, just for the fun of it. And so what we were doing is we thought we would pump tens of millions of images plus the corresponding machine-readable structures into a deep neural network and see what it could learn. The fun thing about this particular approach is we make no assumptions about the underlying problem. There is no design of the concept of bonds or atoms or anything in the design of the neural network. It's a generic combination of a feature extraction network developed by other people, in this case EfficientNet, that was used in image recognition, and a transformer deep neural network, both again generic image analysis tools. and we were interested in what this could do. As I tried to imply, the process is a stepwise process. We start with a 299 by 299 little image of the chemical structure. The efficient net system does the feature extraction, and those feature vectors are then translated into a string representation that is presented to the deep neural network. We've done quite a bit of work on trying to find out what the best structure representation is, machine-readable structure representation. There have been quite some publications already on the question whether smiles are good or deep smiles are good. That's a continuation of the smiles format that is better suited for deep learning. And then there is also a recent development called selfies, which is even better suited for, or was supposed to be better suited for deep learning. And for this particular work, we use selfies, which can be translated back and forth from smiles and into smiles. I'm not going into the details here on the structure of the network, but this is really published by others and being used for other purposes already. So it's well tested deep neural networks for image recognition. We discovered very quickly that we were not able to actually do the work within the lifetime of our, in those days, poor PhD student, because this is so demanding in terms of training time. We were purchasing insanely expensive Tesla V100 GPUs in the early days of Kovulan's work, each of them in those days in the order of 10,000 euros. But it just turned out that the amount of images that we would need to train, 35 million and more, would train in the order of months, just for one training run. And of course, you make mistakes, things have to be repeated, so that's just not sustainable. And very luckily, Google came out with a system called TPU, Tensor Processing Unit, and made those TPUs available in the Google Cloud. And we were able to negotiate a good deal and use those TPUs. If you look at the training times here in the diagrams, you see that for one epoch of training, those trainings go, in our case, up to 60 epochs until they converge. For one epoch, you need 30 hours on a V100 GPU, and you need only a few hours, 2 to 3 hours on a TPU, which essentially means that a model that trains for 3 months on a GPU can now be trained in 11 days on a TPU, and that's a small one. So we could even go to TPUs like a V3 32 TPU that can even train 16 times faster than what we are seeing here. So that was a hardware development that really enabled us to do this work. A few remarks on the training success. So the biggest data set we used was 35 million images. Those were generated using the CDK. We split out a 4 million data set for testing. And you can see here that this is the data set 4, essentially the 35 million data set that we are reaching up to 96% of Tanimoto 1.0. The vast majority of those are isomorphic predictions between the true structure and the predicted structure. We used the Tanimoto because we thought, how big are the mistakes that the network is making? If it doesn't predict the correct structure, how close does it actually get? And therefore we use Tanimoto as a measure for that. But of course, what we are ultimately for this purpose interested in is completely isomorphic and perfectly correct structures. So this system has been deployed, is completely fully open source. It's on the GitHub repository mentioned here. All the data is available on Zenodo, and all the details are in this paper. And Kurulan has spent really a large effort in creating this very informative GitHub site that I would like to point you to. We've just recently made a few extensions. Obviously, we need to get much closer to the partly imperfect depictions that we get from scans from article pages. So what we are currently working on is to essentially screw up the training data a little bit more, so that the deep neural network gets to see real-world data. Obviously, it's impossible to curate this manually, so we have to use automatic diagram generation, but we can use image augmentation to make those data sets more realistic. So here you see a number of images as we can now generate them. Obviously, rotations are important. We have ways to add noise and make things like old scan pages. There are R group labels, arrows in those images. We have atom numbers very often, and so on and so forth. So this is all done using a tool developed by Otto called RanDepict and available in this GitHub site here. And we've recently used, I'm actually not going to show this here. Let's go to the last page. We've just last week taken a Journal of Natural Products publication, ran the DECIMER segmentation. DECIMER extracted those images here. Those are the original images from the publication, and without any changes ran the new DECIMER system trained with these augmented images with noise and so on. And in fact, it was able to translate all of those 5 structures into the completely correct SMILES representations. And that's, I think, of course, as always, a particularly lucky case, but it's very promising that we can now do this. Outlook is that we need to do even more augmentations, even more data, and then see how far we can get in terms of precision. Just 2 more slides. We've tried precisely the same with translations between textual representations of structures. So using images to translate them back into IUPAC names and the opposite direction. And that worked equally well. This system is called STOUT, SMILES to IUPAC translation. It's also published in Journal of Cheminformatics. And with the latest system based on transformers, we can get to really high accuracy of translations between IUPAC names and machine-readable SMILES. It goes close to perfection. Of course, this also has been to be taken with a grain of salt, because it depends on what type of universe of structure representations you are moving in. So how big are the structures? Are you adding stereochemistry and all these questions? Right, and with this I'm done, and I'm happy to answer your questions. Thank you very much for listening.
Great. Thanks a lot, Chris. First of all, Evan says hi. Yeah, so very good people meter. And Saravanan has a question. It's actually about INCHI versus SMILES. So I will read that because I think he doesn't have a microphone. Is the INCHI identifier more reliable or suitable than SMILES, especially to differentiate highly similar molecules? Apart from canonical SMILES, did you try any other format?
We did try INCHI and it performed disastrously bad. It's just not I have no idea why, to be honest, but it doesn't seem to be very suitable for this type of deep learning. In fact, before we used transformers, selfies were a very good representation. Once we moved to transformers, the original smiles format was the best representation, surprisingly, and we are just preparing a small communication about this fact.
Mm-hmm. Yeah, and Roger Sale actually was commenting on that as well. But Roger, if you're here, feel free to comment yourself on that as well, orally, I mean. Yeah, otherwise I will read that. So INCHI will fail to assess bond order assignments. So, for example, Kekulé and tautomeric forms. So that's one of the reasons that Roger is giving you. Yeah. Okay. And Mariam has a comment or question as well. I don't know if you have a microphone, Mariam. Yeah, okay, it seems to be more common. So the latest and most modern of the line notations is INCHI. Okay, it resolves many of the chemical ambiguities, particularly with respect to stereocenters and tautomers. That's right. It just doesn't seem to be that suited for a particular purpose.
But it's just to say, INCHI was developed for a different purpose. It's a chemical identifier. It's not meant to be translated back. It's possible, but it's not meant to be translated back into a connection table.
Yeah, so it's a different purpose, right?
Yeah.
So those will be developed later then in this sense. Yeah. Yeah, thank you, Dimitri. Do you have a microphone? Would you like to ask that orally? Yeah, otherwise I will read that.
So,
yeah, thank you for the very interesting talk. Do you have ideas how to deal with Marcuse structures, which you often find in patterns, for example, as well? What adjustments can you make here?
Yes, I mean, that clearly needs to be tackled. There are, I should not hide that there are alternative approaches also using deep learning for the same system. There was a Russian team who published a similar system, and they have a nice way of dealing with Marcuse structures. Unfortunately, but again, I might be wrong. I think the model is not openly available.
Mm-hmm. Do you work on that yourself as well? I mean, is that something you're intending to address?
Yes, it's definitely something that we need to solve, because we want to solve real problems in information extraction, and also in natural product papers. People often use Marcuse structures and lists of R groups.
Yeah, that's right. Okay, and one last one from Akash. Would it be great to try SMILE's arbitrary target specification, so SMART as well?
It certainly would be great. I don't have any idea how to do this at the moment, but yes, might be a great idea.
Okay. Yeah, very good. Excellent. Thanks a lot, Chris. I think that's very important work. Yeah, also due to the reason that you put structures into PDFs and then need to extract them again. Yeah, so practically right now, unfortunately, that is something that is really needed. Yeah, due to many reasons. Yeah. Okay, great. Thanks a lot, Chris, for your presentation.
Thanks.